Chapter 10, Muscle Tissue, Part 3. Now that we've talked about the basics of the sarcomere, it's time for story time. In story time, we're going to talk about the story of how a muscle contracts. And there are 5 main chapters. First, we have the arrival of the neural signal along the somatic motor neuron. We have the generation of the muscle action potential, that change in charge in the muscle fiber sarcolemma. The release of calcium ions from the sarcoplasmic reticulum, the shortening of the sarcomeres, and of course then the contraction of the skeletal muscle fiber. So here we see the arrangement of somatic motor neurons to skeletal muscle cells. So this is the axon of the somatic motor neuron, the synaptic bulb at the end of the somatic motor neuron that is adjacent to the skeletal muscle cell. So somatic motor neuron in bulb. The place where the synaptic bulb is adjacent to the skeletal muscle cell is called the neuromuscular junction. So neural for neuron, muscular for muscle cell junction because they are adjacent to each other. Now, within the synaptic bulb, we see hundreds and hundreds of vesicles. These vesicles are filled with the neurotransmitter acetylcholine. So these are called synaptic vesicles. Synaptic vesicles filled with acetylcholine fill up the synaptic bulb. And of course, the synaptic bulb is next to a region of the muscle cell that is called the motor end plate. The motor end plate is just the part of the sarcolemma that is adjacent to the synaptic bulb. However, the two cells, the neuron and the muscle cell, do not actually touch. There's a little thin space between them. That space is called the synaptic cleft. The synaptic cleft, the space between the synaptic bulb of the neuron and the motor end plate of the muscle cell. Now, normally the synaptic bulb is always constantly pumping calcium ions out of the cell. So the synaptic bulb has very low levels of calcium ions because it has a calcium pump spending energy, ATP, to send out the calcium ions, causing a high concentration of calcium ions outside of the synaptic bulb and a low concentration inside. Synaptic knob, synaptic bulb, very similar terms. When the neural impulse arrives at the synaptic bulb, it is a action potential. So it is a change in charge. And that change in charge running along the plasma membrane of the neuron activates voltage-gated calcium ion channels. And these open up, calcium ions flood into the cell, going from high concentration to low concentration. When they enter the synaptic bulb or synaptic knob, they will then bind to the synaptic vesicles. When they bind to the synaptic vesicles, this triggers the vesicles to engage in exocytosis, to travel to the surface of the synaptic bulb, fuse with it, and release the acetylcholine into the synaptic cleft. So again, calcium ions flood in, bind to the synaptic vesicles, which then release via exocytosis the acetylcholine into the synaptic cleft. The acetylcholine will then diffuse across the cleft and reach the motor end plate. And when they reach the motor end plate, the acetylcholine will then bind to receptors that are on the surface of the motor end plate. So again, acetylcholine diffuses across, binds to receptors on the motor end plate. The receptors, when filled with acetylcholine, causes sodium ion channels to open. And the open sodium ion channels will allow sodium ions to flood into the muscle cell. So acetylcholine binds to its receptor, opening sodium ion channels. Sodium ion floods into the cell. This is then referred to as a ligand-gated sodium ion channel. Ligand is something binds, in this case acetylcholine. Gated channel, when it binds, it opens the channel, allows sodium ions to flood into the cell and diffuse into the muscle fiber. And that is what generates the muscle action potential. So the opening of these ligand-gated sodium ion channels causes sodium ions to flood into the cell. That changes the charge across the membrane, changes the resting potential, raises it from negative 95 millivolts to something higher, and that creates the end plate potential that then generates the muscle action potential. Change in charge, sodium ions flooding into the muscle. So again, acetylcholine binds to the receptors, opens those ligand-gated sodium ion channels, and sodium ions flood into the cell. Turns out the acetylcholine also binds to potassium ion channels. However, these are slower to open. When they do open, though, potassium ions begin leaving the cell, returning the charge across the motor end plate to resting potential. So first we get a dramatic increase in charge because of the sodium ions, and then the potassium ions slowly return it back to resting potential. Basically, as I just said, reverses the charge difference to make the muscle action potential to be a limited signal, and then enzymes also come in and break down the acetylcholine so that they're no longer in the receptors. So after the end plate potential activates, it causes a change in charge. That then triggers the voltage-gated sodium ion channels to open. They open, sodium floods in. That then changes the charge in that part of the sarcolemma, which then stimulates the next region, sodium ion channels to open, and so on, and it moves down the sarcolemma. Now, this change in charge from the resting potential is referred to as depolarization because it makes it more positive. So sodium ions flooding in from the positive side to the negative side depolarizes that region of the sarcolemma. And that muscle action potential travels down the length of the sarcolemma and into the transverse tubule. At a delayed time, a little after the sodium ion channels open, we have those voltage-gated potassium ions that open. Potassium ions flood out of the cell. They are positively charged, so they then repolarize that region of the sarcolemma. They return it to the resting membrane potential, repolarizing, making it more negative. So again, end plate potential, because of the binding of the acetylcholine, causes sodium ions to flood in. That gives us a depolarized region. That change in charge stimulates adjacent sarcolemma to have its voltage-gated sodium ion channels open. Then those sodium ions flood in, changing the charge in that region of the sarcolemma. That change in charge then stimulates the next region to open its sodium voltage-gated ion channels, and it continues to move down the sarcolemma. And then, of course, following behind is the opening of the potassium ion channels, those voltage-gated potassium ion channels. Potassium leaves, reestablishing the resting potential. Again, end plate potential caused by the binding of the acetylcholine leads to the opening of voltage-gated ion channels that allows a muscle action potential to travel down the length of the muscle fiber. There is a period in which the membrane cannot be activated again, and this is called the refractory period. Short period of time when action potential cannot be triggered. And this is because we have our ions distributed incorrectly. So directly after the muscle action potential moves to the next region, we now have lots of sodium ions inside the cell, lots of potassium ions outside of the cell, and so we can't generate another muscle action potential. Has to wait until the sodium potassium ion pumps reestablish the proper ion distribution. It is also why the muscle action potential can only go in one direction. It cannot go backwards. All right, so the muscle action potential is now traveling down the transverse tubule. Sodium ions are rushing into the cell, and it is causing a change in charge near the sarcoplasmic reticulum. That change in charge causes the calcium ion channels, the voltage-gated calcium ion channels, to open. Calcium ions flood out of the sarcoplasmic reticulum. So again, the muscle action potential traveling down the transverse tubules allows sodium ions to flood into the cell, triggering the opening of calcium ion channels, allowing calcium ions to exit the sarcolemma and flood into the muscle cell. Then what happens is the calcium ion will bind to troponin. So calcium ions that are now in high concentration bind to troponin. Troponin changes shape. When it changes shape, it moves tropomyosin away from the myosin binding sites. And now the myosin binding sites on the actin proteins are exposed. This is the initiation of the cross-bridge cycling. And this cross-bridge cycling has 4 main repeating steps. First up, the myosin binds to the actin at the myosin binding sites. So we now have a physical connection between the thick and thin filaments. This is the cross-bridge. Then the myosin will change its shape, pulling the thin filaments toward the M line. So this is called the power stroke, kind of like stroking done by someone rowing a canoe. The myosin changes shape, pulls the thin filaments towards the M line. This will make the sarcomere a tiny bit shorter. And in this process, ADP and phosphate are released. So the power stroke is the second step. That is then followed by ATP binding to the myosin. ATP binds to the myosin, causes the myosin to release the actin, breaking the cross bridge. And then after that, the myosin will break down the ATP into ADP and phosphate, using that energy to reset itself so that it is in position to bind to the actin once again. So the energy from the ATP is used to return the myosin to the proper shape so that it can bind to actin again and continue the cycle over and over again. Myosin binds to actin, myosin does power stroke, releasing ADP and phosphate, myosin releases actin when bound to ATP, myosin uses ATP to return to its natural shape, and so on over and over again. And this cycle will continue as long as there is ATP in the muscle cell and as long as there is plenty of calcium ions present. So here we have a relaxed muscle, and it has in it the thick filaments, the thin filaments, the Z discs, the M line. You have a nice large H zone, nice large I bands at the ends. So the myofibrils are as long as they can be. Sarcomeres are as long as they can be, with large H zones and I bands. You begin contracting the muscle, and the myosin's pulling the thin filaments toward the M line. As this occurs, it shortens the H zone and shortens the I band. So the H zone and I bands shrink because now the thin and thin filaments are overlapping. And then finally, you have the muscle completely contracted. When it is completely contracted, there is no H zones. There are no I bands. The thin filaments and the thick filaments are completely overlapping. The sarcomere is now basically the size of the thick filament, and this is as much as the muscle can contract. So again, at relaxed state, lots of H zone, lots of I band, only a little overlap between the thin and thick filaments. Start to contract, the myosin's pulling the thin filaments toward the M line. We now have increased overlap, so the H zone gets smaller, the I bands get smaller, and at complete contraction, the sarcomere is as short as it can get, which is the A band, and there is no place where there's not overlap. So no H zones, no I bands. And this relates to rigor mortis in that after someone dies, their cell membranes become leaky and calcium ions leak out, and that causes the muscle contractions found in a recently dead individual. And then after that, the muscles get stuck in that mode because there's no new ATP being generated. So that is how rigor mortis works.